Hello, I'm Dr. Melvin Chua, your guest host of the ASCO Daily News Podcast today. I'm a radiation oncologist, and I currently practice at the Division of Radiation Oncology at the National Cancer Centre in Singapore. I also served as the chair-elect of ASCO Breakthrough Program Committee, and on today's episode, we'll be discussing key takeaways from this year's Breakthrough meeting. The global meeting in Yokohama, Japan, brought together world-renowned experts, clinicians, medtech pioneers, and novel drug developers to discuss scientific innovations and disruptive technologies that are transforming cancer care today. I'm joined by Dr. Lillian Siu, the chair of Breakthrough Program Committee. Dr. Lillian Siu is a senior medical oncologist at the Princess Margaret Cancer Centre and a professor of medicine at the University of Toronto. You'll find out full disclosures in the transcript of this episode, and disclosures of all guests on the podcast are available at asco.org/DNPOD. Lillian, it's great speaking with you today.
Thanks, Dr. Chua. I'm happy to be here.
We were just at ASCO Breakthrough, and it showcased some incredible scientific innovations and really showed us how technology innovations in precision oncology, biotech, and artificial intelligence are transforming cancer care. I mean, what are your thoughts?
Yeah, it was a really exciting meeting, Melvin. The theme of this year's Breakthrough meeting was shining a light on advances in cancer care. And our opening session featured an illuminating keynote address by the renowned thought leader and tech trailblazer Hiroshi or Miki Mikitani, the founder and CEO of Rakuten and Rakuten Medical. In his address that was titled Innovative Technology in Oncology, he spoke so passionately about innovation and really see around the corner to predict what is coming and taking risk. And I think that's what medicine is about, not just what we have in front of us, but to predict and forecast what's coming. I totally was inspired by his address, and I think a lot of the audience felt the same way. He also spoke to us a bit about his company's development in photoimmunotherapy using novel technology and light therapy in head and neck cancer. And I think that's also an area of new technology that we should watch in the next few years.
I mean, I totally agree with you, Lillian. And one of the quotes that he spoke about really spoke to my heart. And I think, he spoke about the two choices and whether to do and not to do, and not to do is not an option. So I think that was a very compelling message to a lot of our audience at a meeting. And then so on this same note, right, innovation is a driving force in oncology. And we saw countless examples of this throughout the Breakthrough meeting. Were there any sessions that really stood out for you?
There were so many exciting sessions. First of all, there is drugging the undruggable session. This is a really important session because in the past, we felt that certain cancer targets, such as KRAS, MYC, etc., are not druggable. KRAS G12C is the poster child in this area. So during this session, we heard about many ways that we are now looking at to target these so-called undruggable molecules in a cancer cell. And we talked about molecular glues, we talked about degraders, and really novel ways that are not yet reaching the clinic called cyclic peptides was discussed by one of the speakers. The other session that is very interesting also is CRISPR and gene editing. Obviously, we all know a little bit about gene editing, really trying to change and knock in some genes that are important, perhaps to change the function. And one of the sessions talked about trail-targeted induced mesenchymal stem cells. And perhaps this is a way to, again, deliver novel therapies and novel treatments to our patients. There were many examples of how CRISPR and gene editing can be ultimately going to the clinic to benefit our patients in terms of therapeutics. I want to highlight another session, which is the adoptive cell therapy. I think everybody knows about CAR T cells, but in this session, we talk about non-CAR T cells or newer things such as off-the-shelf NK cells, natural killer cells, even obtained from cord blood. So this way, it is allogeneic. So in other words, we don't have to rely on only a patient's donation of their samples, but actually get it from off the shelf from other donors. There are other ways to really use human-induced pluripotent stem cells that we can armor them by transgenes and also CRISPR out any unwanted genes, for example, to enhance the effector function of T cells. So many exciting ways to bring these cell therapies to the patients. And last but not least, I want to highlight Dr. Chris Abosh, who is one of our keynote speakers talking about molecular and minimal residual disease and early cancer detection using circulating tumor DNA or liquid biopsy. He talked passionately about the TRACER study, which he is instrumental in terms of leading together with Charlie Swanton in the UK. This is a study that really has uncovered a lot of science about cancer heterogeneity. And in that study, he also studied circulating tumor DNA and really shed a lot of light about clonal and subclonal dynamics over time that changes.
Just to touch on that point about innovation and how that translates to cancer care, I think it was great that we had those case-based applications in lung cancer, in breast cancer, and the viral-associated cancers. And I like how the speakers were able to bring in the yin and the yang, bring the West and the Eastern perspectives at these interactive sessions. I particularly enjoyed all of them, but the session on the lung case discussion, where we know that there were this EGFR mutant lung cancers that are prevalent in this part of the world, in Asia. I thought the interaction between the speakers were fantastic. On the same note about therapies, and we heard about novel therapeutics at this meeting as well, I wonder what your thoughts are, about some of the sessions, and do you think some of these technologies were able to be brought into practice? And your thoughts on the novel therapeutics session that what happened at Breakthrough. Do you think this will actually impact clinical care?
Oh, for sure, Melvin. The 5 areas that were covered during the novel therapeutic session are really drugs already in the clinic. And for example, the first one was about antibody drug conjugates. We know there are now at least 12 antibody drug conjugates already approved by the FDA, and many more likely to be approved in the near future. And the session really talked about what's next, how to improve upon ADC, for example, using better drug antibody ratio, talking about new payloads and really new formats that make perhaps ADCs even more potent in the future. There was a session on oral immunotherapeutics. It was really how to target the innate immunity. And I think novel oral immunotherapeutics is very important because we all know PD-1, PD-L1 inhibitors have been the backbone, but we need another breakthrough. And having oral immunotherapeutics will make it very attractive for patients because they don't have to come to the cancer center to receive the drugs. Another part of that session was about T-cell engagers and biospecifics, really how to bring molecules to the T-cell, to the effective cells, so that they are able to be cytotoxic to the tumor. We talked about also oncolytic viruses. How are the new ways to utilize this kind of natural agent to target the cancer cells? And lastly, we also talked about personalized cancer vaccine, which is obviously very timely. We all know a lot about vaccine now after the COVID pandemic. And how do we use cancer vaccines to be a good therapeutic drug? I think especially important in the earlier disease stages as adjuvant therapy. Some exciting data, for example, in pancreatic cancer as adjuvant really is groundbreaking for this whole topic of cancer vaccination.
That's great. And for me as a radiation oncologist that's not so deep in the drug development, hearing all the talks at ASCO Breakthrough, was really informative for me, and I learned a lot. I mean, in particular, you spoke about the session that was oncolytic therapy and the results in glioblastoma multiforme, we know is a daily disease, was certainly very impressive. And so it speaks to the whole notion that, in fact, some of this stuff are, in fact, reaching the clinic and making a difference in daily diseases. I think there's a lot to take in from there.
Yeah, Melvin, you're so humble. I know you're a big expert in artificial intelligence. And I think the whole session about AI applications in precision medicine really was not just in that session, but a whole theme that went throughout the entire meeting. So I'm very interested to know what you think about some of the presentations around AI and the disruptive technologies in precision medicine, such as next generation multi-omics, etc. What are your thoughts?
No, absolutely, I agree with you. And there was so much material within the AI session, the multi-omics session, as well as the keynote by Dr. Mary Ellen Geiger, which basically speaks about some of the pre-existing work, the historical work on artificial intelligence in radiology. And I'd like first to talk about the keynote by Dr. Mary Ellen Geiger. It was very nice that she elegantly showed how AI was in fact already, in practice in radiology, where it helped to fulfill or to address a need for radiologists. Almost 20 years ago, they were able to show that using computer vision, you were able to basically facilitate the calling of, abnormal mammograms. And it was, inspiring to see how this early thoughts have now basically accelerated a lot of the advances that we see that are in practice today. The other thing that was also nice was to see this global collaboration, the need for global collaboration in the artificial intelligence space, and the radiologists are clearly leading the way. And I think the part of the impetus for this effort came from an opportunity that arose during the COVID pandemic that clearly affected all facets of healthcare. That was a nice segue to the very sort of dense 1-hour session we had on precision oncology care with artificial intelligence. I think when we designed this session, we were very deliberate that we wanted to address all aspects of how AI could be applied, from real-world clinical data. We saw examples of how having good, well-annotated data sets could actually help to accelerate and facilitate liver cancer screening in Hong Kong. Then we also saw a very simple practical application of AI in pathology, where apart from just having this tool to be able to extract features that could potentially predict outcomes of patients and predict drug responses, we saw a very practical example that applying AI in digital pathology could actually homogenize or harmonize the ways the pathologists review their cases. And so I thought that was very neat and could speak to all our clinicians across both developed and developing countries. We also saw very exciting stuff on the use of AI in terms of calling out mutations, because we know that next generation sequencing is pretty much a cornerstone of how we practice in oncology today. And yet we know that there are prohibitive costs that preclude this technology in certain parts of the world. And it was nice to see how AI could actually lower the cost of some of these sequencing technologies, like being used in liquid biopsy. And then finally, there was some fancy science as well that was showcased in the spectrum, where we saw how industry as well as academics are thinking about integrating multi-omic data sets to then be able to accelerate drug discovery, help define patients better, and so that we can think about how to look at precision oncology using targeted treatments for specific patient phenotypes. So I think this was a very nice transition to the multi-omics session, where again, some of these topics were touched on, ranging from liquid biopsies, and this was already covered in Dr. Abel's talk, what you spoke about, and as well as the preceding day session where we heard snippets of it. And it was again reinforced by the speakers when it showcased how liquid biopsies, we've heard so much about it in the last decade, and we've seen it been approved now for use in the clinic, but yet so much remains unknown, like the discrepancies between assays, addressing the cost of assays, and importantly, how do we deal with the information? So I think, we're just at the tip of the iceberg here. A lot of the clinical evidence needs to be generated in due course to address some of these questions. But at the same time, it was also nice to see some of the new technologies being applied in discovery science. So we know that, immunotherapy is a major player in oncology today. And the breakthrough represents a forum whereby we're able to bring translational scientists to showcase their work. And we saw examples of that at this meeting, where single-cell technology, digital spatial technology, being able to apply that in pathology specimens, and how the two are integrated to be able to reveal more novel science to us, to show us how immunotherapy works or doesn't work in some patients. The both of us has touched on so much content that was showcased at a breakthrough. And I think this speaks to the impact, the learning experience we've had from breakthrough. And I think that's the intended purpose of this meeting.
Yeah, I agree. It truly was a very exciting 3 days. And I particularly like the multi-omics session where we see that the technology is so advanced just in a really short period of time. Over the last few years, we've been now able to go into single cell resolution, where in the past, I don't think we would ever dream of being able to do that. In fact, I recall in the single cell session, we can even see messenger RNA on the slide, which I thought was fascinating, something that I cannot imagine we can see by the naked eye. It really is an exciting time in oncology, Melvin, and the field is advancing with these new innovations and therapies. But at the same time, I think it's important that we do live globally, and we need to work really also help to improve access to quality assured cancer medicines and diagnostics in the low and middle income countries. What do you think about that part? Did we do a good job in addressing that in the meeting?
Absolutely, Lillian. That we had a special session that was chaired by Dr. Peter Yu, and the lecture was delivered by Dr. Gilberto Lopes from the University of Miami. And we know that he's a strong advocate for this, and his title spoke to this topic very poignantly about how science, technology, and practice can be enabled in low and middle income resource settings. And I thought that the work that he highlighted, that the whole ATOM coalition, that was important. And ATOM basically stands for Access to Oncology Medicines. And it was established last year by the UICC, the Union for International Cancer Control, along with global partners, to improve access to anti-cancer drugs and to develop processes for ensuring drug quality, delivery, as well as the optimal utilization of medicines in low resource settings. And I think there's a lot more work to be done. Some of the information that you showed was very compelling to me from this part of the world, but we know that Asia is very heterogeneous in terms of the resources, in terms of the culture. And I thought that the drug pricing, for example, how that should be addressed across the different countries is an important topic to pick up. And I hope his lecture only invigorate this conversation going forward.
Yeah, thanks, Melvin. I totally agree. That was very inspiring. And, Breakthrough is such a one-of-a-kind international gathering that we are not only able to network while we're there, we also have a session to really allow attendees to leverage international cancer networks, to learn a bit about them, all the way from, for example, some of the North American groups to Asia Pacific groups to even global groups, and how we interact between pharma and academia, really transcending boundaries. And I think it is really, really important for us to now have these networks address issues such as equity in cancer care innovation, novel approaches, and so much more. And I am sure you're going to do a good job in making sure that gets into the agenda in our next year's meeting in 2024. Ultimately, we hope that these collaborations in cancer research will help to improve the outcomes for our patients with cancer.
Thank you again for sharing the great highlights from ASCO Breakthrough. And I'm really appreciative of your work, your commitment to build a really robust program for this year. So thank you.
And thank you, Dr. Chua. And you can bet that I will not miss Breakthrough 2024 in Yokohama in August next year. I will be there.
Okay, I'll hold you to that. And thank you to our listeners for your time today. You'll find links to all of the sessions discussed today in the transcript of this episode. And finally, if you value the insights that you hear on the podcast, please take a moment to rate, review, and subscribe wherever you get your podcasts. Thank you again.
The purpose of this podcast is to educate and to inform. This is not a substitute for professional medical care and is not intended for use in the diagnosis or treatment of individual conditions. Guests on this podcast express their own opinions, experience, and conclusions. Guest statements on the podcast do not express the opinions of ASCO. The mention of any product, service, organization, activity, or therapy should not be construed as an ASCO endorsement.